Who's absent? Anybody down here? Fix it in this list. Let's pray. I'm just a little bit off today with my last It's got, everything that you should fix things. So we are where we are. But, Lord will help us. Father, please do help us. Help me to teach the things that need to be taught. If there's something we forget, we will just trust that we didn't need it wasn't for us today. Lord, help us. We ask, please, to understand these things and let us see relevance to our lives. Jesus, we pray this in Jesus' name. Amen. All right. Well, this is week 3. Right? Yes. No, week 3. Okay. We have 16 weeks. So we're supposed to do 4 tests. You figure a test after every 4 weeks. So let's figure out when we're going to do our first test. Okay? You got your calendars. It's where you need to be. I want to be mindful if you have something going, it's like I'm not going to be here that day, or I'm getting killed the day before, then we won't do a test. We'll move. Okay. Okay, so this is week 3. That's right. Okay, so next week is 4. So we're looking at probably the 11th or the 13th. 11th, 13th, what do you think? One day sounds better than the other? 13th sounds better? Okay. Biology test. What February? What do we say? 13th. Okay. 14th is Valentine's Day. That's what's good. All right. Okay. So there we go. What's going to be on that test? What do you know on that test? Five characters. Stick life. What else? Viruses. Bacteria. Antibody stuff. Are key. And what else? What are we going to start today? Yeah, that's kind of a side quest. Also part of the side quest. That's all side quest stuff. Where are we going? We're kind of thinking about this. Let's go back to this. Life, right? I'm going to split it right away into 2 domains. What are they? 2 groups. Prokaryote and eukaryote. Okay, and then over here, what do we have? Archaea and bacteria, right? And then over here, I got 4 things, right? First one, Protista. Right here. Fungi. Next, plants. Next, animals. Okay. So, this will be on the test. This will be on the test. This will be on the test. And probably that will be on the test. Let's see. You will definitely cover all unicellular life and likely fungi, which are weirdos. They're strange. They're fascinating. They're strange. Alright, good. Well, let me erase this. what things are going to be on the test? I mean, yes, those topics, but what can you expect on the test? You get much true or false? I think a few, not many. Correct answer to this, everything you're talking about, right? I haven't bogged down too many minutia, right? You know, it's been meat on the bone for class. That's going to stay that way. Okay, so we were looking at antibodies, right? If I reteach something I already taught, you can just hit fast forward on me, okay? Just tell me and we'll hit fast forward. So I believe we were talking about smallpox and cowpox, right? And they're very similar. Very, very similar. And so the things on cowpox, the cell surfaces, or in a case of virus, a virus surface, right? So whatever the the proteins and stuff like that might be on the surface of these things, these have very distinct charges and shapes. Everything in biology is about charge and shape. Very, very distinct charges and shapes. These charges and shapes on the surfaces of bacteria or archaea we refer to as antigens. I'm not giving you the most in-depth view of immunology or of any of us. We're giving you a big, broad overview. Just here we are. We're talking about all these little tiny things that can just wreak havoc on your body. You don't die from it. And maybe you get sick with a cold or the virus, but then later on you don't necessarily get sick with that exact same cold or virus. Why is that? Why do you only get chickenpox once? Why is it you get infected with cowpox and later on you'll be immune against smallpox? Well, it's because of your immune response, because of antibodies, how antibodies respond. So, you get these antigens, okay? These are on the surface of what's called a pathogen, right? So I'm going to draw this thing, I'm going to call it a pathogen. And you might be wondering, what is a pathogen? Pathogen is going to be anything that causes disease. Okay. So this could be a virus, it could be a bacteria, it could be a protist. Be a fungus, an animal. You don't get infected by plants. There are no plant pathogens. So pathogens have antigens. Antigens are simply the surface. What you're concerned about is what this is made of, what its charge is, what its shape is. These are very, very distinct. Okay? Very, very distinct. Your body, if it cannot recognize an antigen, it will have the capacity to defeat it. This is why HIV is so successful. You know what HIV is? It's called retrovirus. It has RNA and reverse engineers DNA within your cells. It infects your white blood cells. It procreates or hijacks your cells' mechanisms to recreate itself. And then it escapes. But when it escapes, it does something very particular. So here's your cell. Okay? Here's some HIV. Actually, I did that wrong. This is more like a processing. So, going back to our viruses, you may remember that they have some nucleic acid and they have some protein. Okay. The protein is there essentially to hold in the nucleic acid and for some kind of a delivery mechanism, typically. All viruses have nucleic acid and proteins, all of them do. If it only has nucleic acid, it's called a viroid. This is affect plants. If it only has protein, it's called a prion. Right? Those are important for animals. But if it has both, it's a virus. There are groups of viruses, however, which have a membrane. They can't make a membrane. They don't know to make a membrane. So where do they get membrane from? The host cell. When they leave the cell, they lysis the cell, they split the cell. And as they go, they take some of the host membrane with it. Okay. This is a fully formed HIV. That makes sense? Did you draw it? Draw it. And what you need to know is that this membrane is the host cell membrane. That's a big deal. My kids like watching videos of animals and stuff like that. Might be like Thanksgiving or something, they want to know about turkeys, so they want to see videos of turkeys. So I'm just pulling up like several videos of turkeys. Well, this one is just hysterical. This guy dresses up like a turkey. I mean, it sounds like a joke, right? Big old grown man. Turkeys are big, they're not that big. Big old grown man dressed up like a turkey, and he's like making turkey noises. He's gobble, 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 and all this kind of stuff. And the turkeys just come right to him. Because turkeys are dumb. And they don't realize that, oh, this is a dude, this is not a turkey, right? He was able to hide in plain sight, and the turkeys never knew the risk. So the guy shoots the turkey with a bow and arrow, right? Why am I saying that? What made that guy effective? Like, I can't just walk up to some, if I just walk up to some turkeys, they're going to run, right? Or attack. They're not, but you know what I mean. They didn't attack him and they didn't run because he looked like a turkey. In the same way, this is your white blood cell. These are the cells that are responsible for destroying things for your immune system. Why don't they destroy HIV?
It looks like a white.
It looks like a white blood cell. It's antigens are your cells' antigens. To be able to destroy HIV on their own, they would have to destroy themselves. That's dangerous. Okay? So an antigen refers to specific markers, substances, molecules on a pathogen's surface, be it a virus, bacteria, protist, fungus, animal, right? And so if you cannot recognize it, you cannot attack it and defeat it. If you can recognize it, but you can't do anything about it, you can't attack it and defeat it. But if you can do something about it, if you can recognize it and then do something about it, you can attack it and defeat it. You've heard of an antibody, right? So let's give a brief little thing on an antibody. I'm going to show you a video, hopefully here in just a little bit. You have cells that are able to recognize antigens and produce what are called antibodies. antibody is going to look something like that. And these are distinct. These are unique. These are proteins. Okay, protein chains. You have different antibodies. You have thousands of different antibodies. Every time you've ever been infected with anything and your body learned to defeat it, it produced an antibody. And it remembers those antibodies, and it remembers how to make those antibodies. And if you ever get infected with that same thing again, it can ramp up antibody production like that. And antibodies are important. They do two major things for you. Number one, they can immobilize pathogens. What do I mean by that? So you take an antibody like this one here, and it just so happens to have the right charge and shape that it sticks to one type of pathogen, or one type of antigen only. Now this is why the cowpox worked with the smallpox. They are so close, they have some of the same antigens. And so when you got infected with smallpox or cowpox, your body made antibodies for cowpox. But those same antibodies also worked against smallpox because they had the exact same antigens on them. So, here's some kind of a cell or a virus or whatever. And so when I say it immobilizes them, it sticks to two of them. It can attach to two at the same time. Well, now, where one was able to infect a cell, two of them stuck to an antibody. It's like they're in handcuffs. They can't do anything. They cannot get into a cell. They're just floating around waiting to be destroyed. Okay? Does that make sense? So, functionally speaking, when I say they immobilize pathogens, they essentially act as handcuffs. Okay? So that's the first thing they do, which is awesome. The second thing that they do is they mark pathogens for destruction. This back end of this is functional to signal your immune system to come and destroy whatever it's holding on to. Okay. If I'm a cop and I'm arresting two bad guys, but there's a whole bunch of other bad guys out there and I need to go get them, what do I need? I need backup. What am I going to do? Get on the radio, right? I'm going to send out an all call for this. And essentially, that's what these function to do. They signal your body, hey, come destroy these pathogens, right? Does that make sense? It all comes down to charge and shape. Every pathogen has a unique set of cell or virus surface molecules with a unique charge and shape called antigens. You produce antibodies that match up, by the way, on this, right? So this part here, those are antigens, right? And this is an antibody. Of course, this is a pathogen. Okay? So pathogens cause disease. Pathogens have antigens. It's just the surfaces on them, right? And antibodies match up with antigens. You're able to lock to it, which will immobilize them and will signal for destruction. This is how your body works. As long as you're able to produce an antibody in time, things won't kill you. Your immune system will be able to immobilize it and destroy it. Antibody in time, you'll die. Rabies. I talked about rabies last time with Louis Pasteur. Traditionally, if you got rabies, you died. It takes about 10 days or more for rabies to kill you. So if you can produce antibodies fast enough,
you'll survive before it kills you. But if you can't produce them within 10 days, you'll die. And so, he got word of a little boy being bit. So he's got 10 days to find a way to help this little boy's body to be able to make antibodies. Now, he doesn't know all this at the molecular level and everything, but just through experiments, he takes rabies virus, he attenuates it, creating weaker and weaker and weaker strains of it or particles of it, and you give the boy the weakest one, and then a little stronger, so the body starts to learn to put together antibodies, recognize little parts of antigens, create antibodies, give him another one, until he's up to a full dose. And now his body within that time frame has learned to make the antibodies, has learned to destroy the rabies virus. Okay? When you go and you get immunizations, and you go and you get vaccines, that's what they're doing. They're giving you weakened forms so that your body will learn to fight it. You take a little baby. Typically you get you have a schedule of these that you're supposed to get. You take a little baby, you go get them their first round of vaccines. What's the rest of the day like for that kid? It's miserable. They feel terrible. You just intentionally put disease in them. They tend to get a mild fever, they're achy, they're whiny, they feel terrible. And it's not because, the shot hurt. I mean, maybe the shot hurts a little bit, but literally they're sick the rest of the day, maybe the next day, maybe the next day. But the goal is that their body will develop antibodies to attack the antigens so that if they ever see the real thing, they're ready. Historically, this has been very important. Things like measles, mumps, rubella, smallpox, diphtheria, tetanus, polio. These things would just kill you. So we just bought it. Are you okay? Any attention? Come on. This is historically and personally just so relevant. We just bought a house in Fairfield, Texas. Okay? What's really unique about this house, and part of the reason we got it at a really great price, is there is a little graveyard 20 feet from my front door. I'm told there are many bodies, but there's only one headstone. And the headstone is double-sided. There's a name on each side. So you look at the dates. The dates, and these are little kids, 12 and 10 years old when they died. The dates on it match up perfectly to the time that a major disease was wiping through the South. Hundreds and thousands of people died. You don't die from measles, mumps, rubella, diphtheria, tetanus not so much, but polio, because you got a dose of antigens. Intentionally, you got a little part, we infected you with little parts of this stuff so that you could develop antibodies so that if you ever came across the real thing, you would already be able to defeat it. Let me show you about that. Can I erase? We're good? Okay. So here's what happens. This is time. This is antibody count. Okay? This is 0. Time 0, antibody 0. You're born, you have no antibodies against anything, right? Then suddenly you get infected. So your body takes some time. Maybe this is the time that you get infected at first. You have no antibodies. Pretty soon your body learns to make antibodies. And it produces. When's it going to stop producing all these antibodies? Do you just produce antibodies for everything you've ever been infected with your entire life? Does that seem like a good use of your cells, of your body's resources? It's not. When do you need antibodies? Huh? When you get sick, when you're infected. So you produce them when you get infected. When do you stop producing them? When you're healthy. So we're going to have some kind of a peak, and then as they start to die off, your antibody production is going to get very low again. Is it ever going to hit zero? No, it's never going to hit zero. Guess what? The way life works, you're probably going to get infected with this thing again. Maybe this is the common cold. You got it once at the beginning of the season. Then, 5 weeks later, you come into class and Ariel's just sneezing all over the place and infecting everybody. And everybody else is going to have to go through this, but you've already been through this. What happens to your body? Well, you already have the memory cells for this. They're already there, they're just hanging out. You don't have a ramp-up time where you're learning. You can instantly go into production. And so your body goes... This is first infection. This is reinfection. This is why if you got infected with the same thing twice, maybe you would be like, oh, I don't feel so good, but you wouldn't have all the symptoms of it. Because your body just starts crushing it immediately. You have to be able to draw that on a test. You want to copy that down, you want to make a giant note and just tell yourself, I must be able to draw and label that on a test. Antibody count versus time, first infection, second infection. That little boy in the box, this was his response. Later on, when he got infected with smallpox, this was his response. He already knew how to defeat it, his body came in and crushed it. When you take your, you get infected or injected with what is wrong with it? With vaccines, your body's learning this. Do you have any questions for me about this? Anything you don't understand? You want to know more about it? What's the difference between an antigen and a pathogen? Okay, what is a pathogen?
It's a disease causing.
Yeah, anything that causes disease, pathogen. Pathogens have antigens. Are pathogens the only things that have antigens? That's a good question. No, in some sense, everything is an antigen. How about pollen? Pollen. Anybody get like crazy allergies? I get crazy allergies. I have to take stuff year round. I've got sprays, I've got pills, I got everything. It's because antigens on the surface of pollen. And so it causes an immune response. You ever heard of Crohn's disease, or Hashimoto's disease, or where's some of the others? The one with the wheat that everybody gets. I have no idea what I'm talking about. Okay. Yeah, well, it has to do with gluten, but there's a particular name for it. I can't think of the name of it. It's an H. Anyway, these are situations where your body is attacking itself.
autoimmune diseases. Your body is looking at antigens on its own cells and flagging them for destruction. Or your body is presenting things like with various gluten allergies. Your body's taking wheat and going, here, this doesn't look right, destroy us. And so every time you eat bread, your body's going, hey, destroy these cells. And it causes inflammation, all kinds of problems. This is not good. Okay? So antigens are more, they're on much more than just pathogens, right? Definitely every pathogen has antigens. What's the difference between an antigen and an antibody? Or what's the relationship between an antigen and an antibody? Antibodies stick to antigens based on 1, 2 words. Charge and shape, charge and shape, charge and shape. If you had me last semester, you know those 2 words. We talked about them all. Day 1 in the first semester was everything in science and biology about charge and shape. When we get to the last day of the semester, that will still be true. It's autonomous. Your cells are amazing. You are amazing. How God is engineered. Okay. Well, let's roll on. We're going to go pretty quickly through Protists. Okay. Protists are, I haven't told you this yet, they're the junk drawer of eukaryotes. Said that? What's in a junk drawer? Speak. Junk. What do you mean by that? You have a junk drawer? You have a junk drawer? My grandmother had the best junk drawers. She always had this one. You go into it, there'd be, my grandfather worked for the post office, so there'd always be like 100 post office pens. You know he was stealing those from grandma. There would always be 85 rubber bands. There was usually like a little mini hammer. Why they made these little, you know what I'm talking, they're like little tiny, like why does this exist? It might be broken electronics, and why were they in the junk drawer? Why is stuff in your junk drawer? Just in case. Why is it in your storage drawer? Because what's in your storage drawer? So, why isn't it with the plates? Because what goes in the plate cabinet? Plates. Why isn't it in the fridge? Because what goes in the fridge? Food. So you've got this drawer that you're like, this doesn't go anywhere else, but I don't want to throw it away, so I'm going to put it here. So when I say that protists are the junk drawer of eukaryotes, this is what I mean. They're not animals, they're not plants, and they're not fungus. And they're not prokaryotes. So what are they? Yeah, what are they? What are they? What do you know about them? They're what? They're not multicellular. No. Not multicellular. They do have a nucleus. What else do you know about them? Eukaryotes that have nucleus. What makes a plant a plant? Does a cell wall? What else? Photosynthesis. Good. What makes an animal an animal? They eat. What else? They move. What else? If you're going to move a lot, you can't have a cell wall. Right? Ready? This is a tree. This is a tree doing photosynthesis. This is a tree running a marathon.
This is a tree in love. This is a tree giving a hug. Like trees don't really move, right? This is cell walls. Animals have cell walls. They can move and all kinds of stuff. So, what makes a plant again? Give me at least 3 things. Give me 2. Give me 2 again. Cell wall, photosynthesis, and one more. Multicellular. Because guess what? I've got protists that have cell walls and do photosynthesis, but they're not multicellular. So some of these, some are plant-like. What do I mean by that? Photosynthesis and cell wall, right? Some of these are animal-like. What does that mean? What do? They eat and they... Yeah? What do they not have? Yes, that is correct. Sorry, I shouldn't go with that. What helps them to be able to move readily? No cell wall. Okay. So animal-like, they eat and they have no cell wall, / they move, right? Okay. All right, and some are going to be like what? Fungus-like. Well, you did really, really well with plants. You did really, really well with animals. So what can you tell me about fungus? They do have cell walls. So some of these may have cell walls, some may not. What do fungi do? They do eat, but what do they eat? These are decomposers. Yeah. So, you made a very good point, you eat. These guys both eat, right? And you made a point about these guys. You said not only do they eat, but they hunt. I should have made more of that. That was good. So these guys hunt. These guys don't hunt. You come into contact with fungus a lot here in East Texas. You grew up in Las Vegas or something like I did, you don't. They're there, but you don't notice them. Tell me about some fungus here. Not moss. Mushrooms for sure. Where do you come into contact with this? Where do you see them? In the grass? Where else do you see them? In the woods? Where? Trees. Lots on trees, right? Dead trees or dying trees. You see a tree covered up in mushrooms and stuff. That's a problem for the tree. It's a sign that that tree is not able to fight them off anymore. You don't see a good, young, healthy tree covered in fungus, right? Okay. You definitely see them on dead trees like crazy, right? If a tree hits the ground, it won't be long. And you know what? If you come back after time, what's going to happen to that tree? It's going to be gone. When we say that these are decomposers, what are they doing? They're breaking it down, they're recycling it. Why? It's very, very important. Given the age of the earth, and I don't, for this, I believe in a young earth. I believe in a biblical account of the age of the earth. Whether you believe in that or whether you believe the earth is 6.2 or whatever billion, it doesn't matter. Either way, given the age of the earth, the entire should be covered in leaves. Like every square inch of solid ground should just be covered in leaves. But it's not. Where do they go? Huh?
Yeah.
Cut them down. Decomposers. Fire would do good, right? A little lightning strike would burn them up. That works some, right? But that's not nearly as significant, or as significant a role as decomposers are. Mushrooms, fungi, you hardly notice them. You only really notice them when there's, in fact, you only notice them or see them when they're reproducing. That's by the way, is what a mushroom is. They're sex organs. They produce little spores and reproduce out of those. Yeah, fun fact, let's do this. I got mushroom 1, I got mushroom 2. They run into each other. When do they run into each other? When they're out of stuff to decompose. Okay? I run out of food. I stretched out this way. I was going as far as I could, because they just keep growing. And I was running this way as far as I could. We ran into each other. I know I can't go this way and expect to have food, and I know I can't go this way and expect to have food. Times might be about to get tough for both of us. Let's reproduce. And so they begin to fuse, and they produce a fruiting body called a mushroom. Well, these are called mushrooms. They're not all mushrooms. We'll learn more about that later. And this thing produces little spores that hit the wind. And then that mushroom lands somewhere else and it begins to grow. And you'll never see it. You'll never see it until it runs out of food, and then it'll produce another mushroom. You only see mushrooms when times are tough for the mushroom. As long as they're doing well, you'll never see them. Okay. That's sex, by the way. Okay. So, all of that little sidetrack was because we're talking about the idea of decomposers. They're very, very important. Okay? So, protists. Things you know about protists, things you need to know about protists. They're not multicellular. They do have a nucleus. Modes of nutrition or motility or whatever else. Some are plant-like. They do photosynthesis. Some are animal-like. They hunt, they eat, they have no cell walls, they move. Some of them are more fungus-like, they're decomposers. You ever heard of a slime mold? Cellular slime mold or plasmodial slime mold? Let me pull one of these up. These are some of the coolest things. These glow in the dark. They're on trees and stuff in the right places. And if you film them at night, they glow in the dark. And if you did it on one of those like time things where it's like you speed it up, time lapse order, they pulse. It's crazy cool. Give me just a second, let me pull this up. I can't run a machine on this at the same time. Well, goodness. I guess we're not looking at plasmodial slime mold. I'll show you that next time over. Okay. Well, let's go back to our protists then. I'm going to erase what I got up here. I got a question for you. How do I classify something that doesn't seem to fit? Because all I've said, like, really is it's not like all these other things. And so how do I begin to classify, how do I begin to organize or put some method to something like a protist? Is it shape? Is it color? Is it size? Is it how it moves? Is it how it eats? It's the most, when I just tell you this, I haven't looked at how protists are being classified in many, many years. Last time I saw anything about it was at least 10 years ago when I was teaching AP biology, which is the high school equivalent of this course. Okay? Using Campbell's book and all, and it had a classification. I was very familiar. It was very comfortable for me. I look at this and I go, well, that's very different than anything I've ever known. And then actually, the one 10 years ago was fairly different from the one that I learned when I was in AP biology in high school. Because over time, I'm 45, am I 45? I'm 45 years old. I was 15 when I started taking AP biology. So that's 30 years. So over 30 years, how scientists have classified protists has changed dramatically. At one point in time, it was as simple as this. We called them plant-like, animal-like, and fungus-like. We looked at literally like how they got their nutrition. Scientists no longer look at it like that at all. It's based on their DNA, which breaks down into all these things. It's very, very complicated, minutiae on stuff that you don't care about. You just don't. I am not going to bog you down with memorizing all the different phyla of these. Okay? You don't have to know Chrysophyta and Phaeophyta and Rhodophyta and all these kinds of things. Phyta would be good to know. Do you know what phyta means, or phyta? It means plant. That's it. Do you know what zoa means? Like if I said protozoa? Yeah. So protozoa is like before an animal. It's like a little pre-animal. You don't have to know that. But anyway, things you do need to know about these is they're prokaryotes, right? Yes? Or I'm sorry, they're eukaryotes, right? They're eukaryotes. You must know this. What must they have if they're a eukaryote? You must have a nucleus. Is that the only thing? Is there anything else that eukaryotes have? Not necessarily, because these guys do not.
Membrane.
Membrane bound organelles.
Yeah.
Yeah. Membrane bound organelles. These guys have membrane bound organelles. In particular, they have two that we're concerned with. They have more than two, many more than two, but there are two in particular that we're concerned with, and they are mitochondria and chloroplasts, or plastids. I'll say that. Say it that way, plastids. So there's more than just chloroplasts. Chloro means green. So they're green plastids, but there are other colored plastids. Chloroplasts are green because of what molecule? Say chlorophyll. Chlorophyll is a pigment. Say pigment. What's a pigment? Something that absorbs some color and reflects others. Your shirt is a nice baby blue. Why? What color is it reflecting? Nice baby blue. What's it doing with all the others? Absorbing them. Yours is purple. Why is it purple? What's it doing with purple light? It's reflecting it. What's it doing with all the others? Absorbing it. Chlorophyll is green because what's it doing with green light? Reflecting it. What's it doing with all the others? If you were sadistic and wanted to kill a plant, shine just green light on it. Can't use it. Be mean, right? You ever seen plants that are not green? What are some other colors you've seen in plants? Like a maroon color, a purple color, a brown color? Because there are other pigments that plants can use to do photosynthesis. And that's definitely true with our friends the protists. Okay, it's not just chlorophyll that they can use, though they can use chlorophyll. So these guys have membrane-bound organelles. What do I mean by that? Well, there must be non-membrane-bound organelles, right? There must be. In particular, the things that prokaryotes had. I'm only going to worry about one of them. He said non-membrane-bound organelles. The one here, the one we really worry about, is a ribosome. Remember what a ribosome is? Ribosomes read RNA and make protein. Ribosomes are great, ribosomes are cool. If you're going to live, you must have protein. If you're going to have protein, you got to make protein. If you're going to make protein, you must have ribosomes. So of course bacteria and archaea have ribosomes. They have to. But who else has their ribosomes? Point at yourself. Yeah, they have it. Just because we say they have membrane-bound organelles doesn't mean they don't have non-membrane-bound organelles. They have those too. Anything a prokaryote has, there's no reason to think that a eukaryote doesn't have it or can't have it, okay? But if it's a membrane-bound organelle, these guys do not have it. In particular, mitochondria and plastids will focus on chloroplast. So I want to show you some structure for these things, okay? So mitochondria, this marker's getting a little rough. Mitochondria tends to look like this. It has a double membrane system. I've got an inner membrane and I've got an outer membrane. I'm not going to take you through all this, but the purpose of that was so that you can create concentration gradients of hydronium ions so that you can power ATP synthase so that you can generate ATP. That's why the mitochondria is referred to as the powerhouse of the cell. Okay. So this thing has a membrane-bound organelle. It's a membrane within a membrane. That's crazy and cool. A chloroplast has stuff like this. If I had more markers, I would have drawn that in green because it is green. Now a green marker. This is where your chlorophyll is at on these thylakoid membranes. Again, these are membranes and that's a membrane. This is a double membrane system. Of course, there are many more types of organelles that are membrane bound. These two are double membrane bound. There are other things about these that are very, very fascinating. This thing has its own DNA. This thing has its own DNA. These are self-replicated. They have their own DNA, distinct from the rest of the cell, and they can replicate themselves. Things they cannot do is survive on their own. You take a mitochondria and put it with everything it needs for life, and you just have it sit there, it will die. You put a chloroplast, and you take it out of a plant cell, and you put it there with everything it would need for life, it will just die. These things are not autonomous at all. Now, this is interesting. According to the theory of evolution, how do you make the jump from a prokaryote to a eukaryote? The thought is something called endosymbiosis. Endo, what does that sound like? Not bad. Endo means inside. Well, symbiosis. Sim, we had that one the other day, or sin, didn't we? Together. Bio, life. So life together. So inside, life together. It's the idea that one cell ate another cell, but instead of eating it, it just became best buddies. And so theoretically, you get maybe some little prokaryote that eats some animal-like prokaryote that absorbs a plant-like prokaryote, but instead of eating it, destroying it for food, they just hang out. And it provides the plant-like one with everything it needs, and the plant-like one provides it with carbohydrates, and they hang out their best buds forevermore, and we make the leap from a prokaryote to a eukaryote. Of course, we still have to get the nucleus, but. Or conversely, you take one and it's got a little buddy that, this guy's just amazing at making energy, but it can't defend itself or do anything else, and we absorb it, and we hang out, and I feed it, and, that's the idea of endosymbiosis. Write that word down. Endosymbiosis. If you know what each part means, you know what it means, right? Endo means inside. And you guys did great. I was very proud of you. You knew what these meant. Sim means together. And bio, or biosis, life, right? So inside, life together. You think about that and all of a sudden it should pop. This is a theory of evolution that says that one cell engulfed another cell and they lived together instead of just destroying it like it otherwise always would have. So this is their theory to bridge the gap for how you get a mitochondria or how you get a chloroplast. Me, I'm not buying it. We don't see this anywhere. And this cannot survive. And this cannot survive. They would go, hey, well, this has DNA and it's self-replicating. This is DNA and it's self-replicating. You go, well, to me, that's God's design. That's incredible. I don't have any problem with that. All right. This is important. One of the reasons that protists are important, one of the reasons that your textbook spends time on it, and frankly, so much of what it spends time on is how these are viewed evolutionarily, right? These are thought to be a bridge between something like a bacteria and you. Okay. All right. What else do you need to know? Let's talk about certain ones. You realize you're getting a very curated version of biology, right? Okay. Let's do chlorophyta.
Chloro means? Chloro means? Green. And I told you a minute ago, phyta means? You ever heard the word neophyte? Usually it's to insult someone. You call somebody a neophyte. Neo means new, phyte means plant. In other words, like in the scheme of life, you're not some great big tree, strong and tall, you're like a little baby plant. It's an insult. It's a very intellectual, nerdy, wordy insult. Green plant. Phyta means plant. Green plant. Are they plants? Are they plants? Are these protist plants? If they're plants, where would we put them? With plants. But they're in the junk drawer. They're not plants. This is what we call a misnomer, a false name. Okay? So green plant is what it is, or what it's called, but it's not what it is. We call it that because it's green, and we call it a plant, though it's not, because what does it do? Photosynthesis. So this name does tell you a lot about it. These things are green, and they do photosynthesis. There's only one that I'm really worried about you knowing about, and it's called Volvox. And if I brought my laptop and everything was working right, I would show you cool stuff about Volvox. Volvox are really neat. They're colonial. What does colonial sound like? Colony. What's a colony?
Yeah, you live together.
Is a colony huge? No. No, a colony's relatively small. If it were bigger than a colony, you'd call it a town, right? Or a city. But it's more than one person, right? It's a group of people. So same thing with a colonial protist. So Volvox exist. They can't exist by themselves. They have a little flagella. Here's a. I didn't get a green marker. I don't have one. So let me give you a little Volvox, okay? I'm going to give you some chloroplasts. I'm going to have chlorophyll, okay? Chloroplasts. I got to give it a nucleus, right? So there's a nucleus. It's going to have a mitochondria. I realize you're not going to draw this, but that's okay. All right, there's a Volvox. Kind of looks like sperm cells, right? It's got a flagella. What does that tell you? It can move. So it's plant-like, but it can move. Right? It's also going to have a cell wall, isn't it? That's weird to think about something with a cell wall that moves. Anyway, so there's a single Volvox. But they're colonial, which means will I normally ever see them with just one? No. But because they're a protist, it can live with just one. I could take one of these, get everything it needs for life, come back a month later, and it won't be just one anymore, will it? It will become many. How's it going to grow from one to many? It's going to do photosynthesis. That's how it's going to grow, but how's it going to get from 1 to 4 to 8 to all that? What's it going to do? Ready? Show me. Binary fission, right? Over and over again, reproducing itself. Okay, so Volvox will look like this. Where each little dot is an individual Volvox. It's a colony. This colony is not round, it's a sphere. It's like a little balloon of Volvox. Anybody? Anybody? There are more Volvox. Where are they? Inside the bigger Volvox. If this is a colony, these are daughter colonies. As this colony gets big, it's already preparing for the next generation. It's already producing a smaller colony within itself, and as it gets bigger and bigger and bigger, and this gets bigger and bigger and bigger, eventually this will just leave, and it'll form another little colony. These things are cool, and when you look at it, these live in like ponds and stuff like that. It's microscopic, right? You're not going to see this with your naked eye. But all of a sudden you look under a microscope, and you're like, whoa. And these things, they float, they move. What causes them to move? The flagella. They can can coordinate the movement of the flagella, and they can rotate. And why is it useful? Why would you want to move? Yeah. How do they get their food? Green plant, right? So they do photosynthesis. So it's not to hunt. Why else would I want to move? Yeah, maybe to get away from something that's hunting. Or what if I'm in the shade? Maybe they're hunting, but they're not hunting food, maybe they're hunting sunlight. So we're going to move from the shade or from the dark to the light. So movement is a good foundation that God has given them to be able to function. Okay? So this is a volage. Another really important one of these would be kelp. Kelp's incredible. You've been to the ocean, to the beach, and you see some kelp wash up, and there's this big old like ball on a blade. You ever seen that? Huge. This is a real thing. You can pump that. Did you think that a kelp was a plant? Of course you did. Why would you not think kelp is a plant? What does that look like? It looks like a plant. I see leaves, I see a stem, I see roots. That looks like a plant all day long. It's not a plant. See, a plant, if I just took one random cell from any spot, it's going to die. It cannot live, it cannot reproduce. But I can take one random cell from anywhere on this thing, set it free in the ocean, and it will reproduce, and it will make all of this. I can take a cell from down here and put it here, and it will become part of this. I can take a cell from here and put it here, and it will become part of it. It's not multicellular, okay? Though it has tons of cells. The idea of being multicellular is you must be multicellular. Those cells are highly, highly specialized and cannot live apart from the whole system. That is not true of kelp. Though, oh my goodness, is this getting pretty sophisticated, right? So parts of the kelp, you got the holdfast. It's not a root, but it's kind of like a root. A root gets nutrients, right? It's water. This is not, all this does is anchor me to a rock so I don't float off. This is called a stipe. It looks like a stem, but it doesn't function like a stem. In a stem, I'm taking sugars down and I'm taking water up. This does not do that. These are called blades. Blades look like leaves, don't they? But what leaves do is they receive water from the roots and up through the stem, and they lose carbon dioxide. They absorb carbon dioxide and let out oxygen and water by opening up. These don't do that. These do photosynthesis though, but so does that, and so does that, and so does that. Every single part of this does its own photosynthesis. So the whatever specialization they have is not in terms of function, like cellular function. They're all cellularly functioning the same way. The only specialization they have is where they're located, and maybe the superstructure. And then you come to this, and this is pretty cool. This is called an air bladder. It is exactly what it sounds like. It's thick, and it's hollow, and it's filled with air. Why would I want to have something thick, hollow, and filled with air attached to my leaves? So they float. Because it will do me no good if all these lay down. The surface of these do photosynthesis, but this won't.
And if this lays down over this won't do photosynthesis, right? It's similar to how branches on a tree are held out this way so that all the leaves hopefully get exposed to the light. That's what this is helping these guys to do, is to stay horizontal, okay? So you got a scythe, you got a holdfast, you got a blade, you got an air bladder. Every single cell here does photosynthesis. They're not functionalized, specialized in terms of cellular activity. They all do the exact same things. The only thing specialized is where they're located in the larger superstructure. All right, so we talked about Volvox, which is a chlorophyta. These are part of what's called phaeophyta. It means brown, which I always think of pheo in Spanish as ugly, right? So I always think of these are pretty ugly. It's an ugly brown. It's not a pretty brown. Another one that's kind of cool is rhodophyta, red phyta. There's one in particular, it also has to do with the ocean. It's called dinoflagellata. You ever heard of this? Do you know what the prefix dino means? You learned about this when you were like 4. Dino. Huh? No, that's archae. Archaeology. Dino. What words do you know that have dino in it? Dino. Dinosaurs. What does dinosaur mean? No? Something about a lizard. Terrible lizard.
That's what it is.
Okay. So dino means terrible. Flagellata means it has a flagella. And actually, dinoflagellata has 2 flagella. Dinoflagellata causes what we call red tide. It's the mascot, or the name of the mascot for the University of Alabama. Do you know? I have no sports fans here at all. Huh? Crimson Tide. What does crimson mean? Red? Red. I'm trying to connect. I'm just trying to give you guys anchors to things in the world that maybe you'll know so that you can remember these. So, Crimson Tide is the mascot for Alabama, although they actually have an elephant, but they call themselves the Crimson Tide. Their whole thing is to go roll tide, roll tide. You never heard of this? All right, anyway. Okay, so red tide is a condition where these guys were in the ocean. They're always in the ocean. These guys are in the ocean, and there's an abundance of nutrients. That's a whole other thing. How do those abundance of nutrients do? There might be pollution. It might be upwelling. Maybe a cold current came and brought up nutrients from the deep. And when times are good, what are they going to do? Huh? Multiply. Yeah, like crazy. Make lots of us. And then we use up all the nutrients. This is an important graph. We need to get this. This is just classic. I think I want you to know this one also, okay? I don't think I know. Now I gotta draw it right, but. There's gonna be time. There's gonna be quantity. Okay? We're gonna have 2 things on here. That's good. That graph is good. You need this graph. This is an important connection. This is a principle that we see all across the world in all kinds of things. I got two lines, right? There's a relationship between the two lines, right? What's the relationship? Those are my, my x is time, my y is quantity. What's the relationship? Let's call the black line A and the blue line B. What's the relationship between A and B? Similar.
They existing at the same time?
Say it again.
They existing at the same time?
They exist at the same time. What's the relationship between them? As blank, so blank. What's the relationship?
As A decreases.
As A it decreases, then what?
B increases. B decreases.
Let's say it again. Start again. As A what?
Increases.
As A increases, then
B increases.
B increases. At the same time?
No.
That's a little bit of a lag, right? And then as A decreases, B
decreases.
Yeah. But it did, look at how A just kind of went back down to normal. Maybe had a slight dip and then came back to normal. What happened to B? B crashed and then had to come back up, right? So as A rises, B rises, but then as B rises, what happens to A? As B rises, A gets used up, right? And so A quickly goes down. Well, then as A goes down, B crashes. When we're talking about dinoflagellata, and I said the whole thing here is red tide happens because I got these dinoflagellates in the ocean. They're hanging out all the time, but suddenly there was an increase in nutrients. Which one's nutrients, A or B? A is nutrients. So the B must be dinoflagellates. So when my nutrients increase, the population will increase. But as the population increases, the nutrients get used up. When the nutrients go away, the population crashes. There's not even enough nutrients to sustain the original population, right? So we're going to come down lower, and we've got to slowly come back up. This is a boom-bust cycle. This happens in nature. This happens in ecology all the time with all kinds of organisms. Okay? So what happens, though, for red tide is when these die, they rupture. When they rupture, they release endotoxins. What does endo mean? Inside. The toxins were inside. They weren't harming anybody. These are in the ocean all the time. Every time you've ever been to the ocean, anywhere in the world, these were around. They never bothered you. They never caused a single problem. But when the nutrients for these guys go crazy, their population goes crazy, they use all the nutrients, they crash, when they die, they rupture, they release their endotoxins. What does the word toxin mean? What's it going to do? Huh? It's going to poison things. One of the things, so red tide. Why is it called red tide? Because it's red. Because quite literally the water will be red. Okay? You'll see it. When the water's red, you know what else you'll see all over the beach? Dead fish. Fish will be dead by thousands washed up on shore. Never eat a fish that's dead that's washed up on shore. Ever. Ever, ever, ever. You eat a fish during, fishermen who do like troll fishing or net fishing or whatever, you can't do it during red tide. You capture fish during red tide, those fish are taking in all the toxins from the water, they'll kill you. Red tide will kill people. Okay? This is why we're talking about this. This is one of those things that's just important. If we lived on the Gulf, on the coast, I guarantee you in high school or college on the coast, they're talking about this a lot more than we are. Is it time to go? Now they're all kinds of tired of it. Oh, it's 5-6 minutes? Okay. They also glow in the dark in bioluminescence. So, like if you go to the beach at night and the waves are going and you see it glowing, it's from dinoflagellati. All right. I'll see y'all Thursday. God bless.